Hello, everyone. Welcome to Human Anatomy and Physiology 1, the first term of Anatomy and Physiology at Clackamas Community College. My name is Christina Wallace, and today we're going to talk about our last lab report, the special senses, which will cover vision, hearing and equilibrium, and taste. Okay, here we are at the lab online instructions for special senses. Let's start with vision. So on page 1, we're going to start with the muscles that move your eyes, and then you're going to go on the bottom of page 1. There are some links to videos that go through a cow eye dissection that's incredibly informative and interesting. The first video is the external features of the eye, and the second video is the internal features. Same team of people. Then we go on to page 2 and 3 is kind of a learning about structures of the eye and vocabulary functions for the structures that we just learned. And then page 4 is going to go over a diagram of the visual pathway to the brain. So here we are at your lab report. So the first page is really just kind of about eye muscles. And then, so you have 4 straight muscles. Rectus means straight. And so they kind of come straight from the back of the eye forward. And then you have 2 oblique muscles, which are diagonal. The functions of the rectus muscles is pretty intuitive and straightforward, but the obliques are a little bit counterintuitive. So you can check out page 556 in your textbook to read more about how those muscles work. Then continuing on, you just look in the mirror and move your eyes up and to the left and up and to the right, follow the directions and figure out what muscles are working in the right eye and the left eye. And then the bottom of page 1 has links to the cow eye video or cow eye dissection videos. First part internal or external features, second part internal features. Then at the bottom of page 1, use the following page to label the structures of the eye and identify structures. And so next page is just all about structures of the eye. So in my slides, I've given you some diagrams of muscles. Here are the all of the 6 eyeball muscles. And so you have the nose indicates the medial side of the eye on the far side. So this is the lateral side of the eye. So this would be a right eye. And so you can see kind of the superior rectus, lateral rectus, inferior rectus coming back straight. And then this diagonal muscle here, inferior oblique, and then the superior oblique starts at the same origin point, and then it moves medially towards the medial side of the eye orbit and attaches. It goes through a little sling made of a ligament, kind of dense connective tissue, and then it connects to the back of the eyeball. Here's a front view of the eye with the superior, lateral, inferior, medial rectus muscles and the 2 oblique muscles. I liked this diagram because it also indicates which cranial nerves serve which muscles. And then because you can't see a nose or anything in this picture, think about how you can tell if this is a right eye or a left eye. All right, so structures of the inside of the eye. This picture, straight from your lab book, talks about the tunics of the eye, or layers. So tunics or layers. There's kind of 3 overarching layers of tissue in your eye. So the first one is the fibrous tunic. And so the white part of your eye right here, and then the clear cornea right here is fibrous tunic. And imagine the muscles have to attach to this layer. And so it's your eyes are not as weak and jelly-like as they might appear. The fibrous tunic on the outside is very tough. And because it's tough, dense connective tissue, it's avascular. And so it doesn't have any blood vessels in it, per se. And so the second layer is the choroid, or the vascular layer. The choroid is part of the vascular layer. So the vascular layer includes this red layer and the iris and the ciliary body All of this stuff is part of the vascular layer, and it's the blood vessels of the eye. And so this vascular layer helps to bring nutrients to the fibrous tunic and to the retina. And so then let's see, the third layer here is the nervous tunic. So this is the retina and the optic nerve. And this is where the photoreceptor cells, the sensory cells of your eye live. And imagine your retina is kind of like a big IMAX screen that is pushed up against the back surface, posterior surface of your eyeball. And so we need something to hold it in place. And so that's what this jelly blob is. Do you see that there's this jelly blob in here? That jelly blob is called vitreous humor. And it's a blob of jelly that gives your eye some strength and shape, and it also helps to hold your retina onto the back of your eye. You are born with all the vitreous humor that you'll ever have. In the front of your eye, up here, like in this part right here, and circulating around the lens, you have aqueous humor. So there's kind of a division of that part of the eye. And these little muscles down here, or little processes, make liquid that circulates around the space between the cornea and the iris, and then around the lens to clean and moisturize and bring nutrients. All right, so back to your lab report, just label this picture. And then this is kind of, here's your fibrous tunic, your vascular tunic, and all the pieces, parts of it. The nervous tunic and all the pieces, parts of it. I want to draw your attention to optic disc and macula lutea and fovea centralis. These are areas of your retina that either have no photoreceptors for the blind spot or have heightened condensed photoreceptors, the macula lutea and fovea centralis. And I have some pictures to show you about that. And then on to the eye structures, page 3 is just kind of learning the functions of all of these things. And I have some pictures in my slides that will help understand some of this stuff. So first off, about those retinal structures, the fovea centralis. If you imagine light coming into this eye, so let's draw some light coming into the eye. I'll make the light a blue color. So light comes into your eye, it kind of goes in a straight line. It comes in like this, right? If you see, you can tell that the optic nerve is more medially located. It's closer to your nose. And so it doesn't exit off the exact back of your eye. But if light is coming in your eye and hitting directly, that's the clearest, easiest path. Well, isn't that handy that it's very close to the macula lutea and the fovea centralis? And so if we draw a straight line from the light coming in and it hits, this is the area that it's going to hit the most and the best. And so fovea centralis is going to be your area of highest, most acute vision with more color cones. And then your optic disc, conversely, is an area that's right where your optic nerve exits. And this is going to be an area with no photoreceptors, no ability to send signals. So we'll see that in a minute. I have some pictures for you. All right, let's just take a brief look at your structure of your retina. Your retina has 3 layers of cells. And so you have ganglion cells right here. Ganglion cells in this row, bipolar cells that are these pink ones kind of in the middle, and then in the back are the rods and cones, the actual photoreceptors. And so light is going to come into our eye in this direction. It's going to hit the rods and cones and stimulate those cells, and then we're going to have the signal come back out through the bipolar cells and the ganglion cells, and then we'll start to gather all the axons together, and it'll leave as a optic nerve. So yellow light coming in, exciting the photoreceptors, signal being sent back out through the ganglion cells into axons. So remember that fovea centralis and the optic disc that we talked about. Now that we've seen the structure of the retina, it'll make a little more sense about why these structures do what they do. You don't have to know this. You do not have to know the histology. I just put these pictures in because I think they're cool looking and it helps to kind of explain, in my view. All right, so first off, just notice this. This is a cross section of an eye. And so the open space in the middle is where all the vitreous humor would live. And then the retina is pushed up against the back posterior surface of the eye. Here is our optic nerve exiting. Here is our clear cornea. And then here's the lens right there. So I just kind of wanted to see, this is what we're looking at, this little square piece right here of the optic disc. So you can see the layers of the retina right here, the layers of the retina. So you have ganglion cells, bipolar cells, rods and cones, right? And so the signal comes in, it goes to the rods and cones, and then it gets kicked back out and goes to all of these ganglion. And then the ganglion are going to all exit through the optic nerve. Does that make sense? All right, so that's why, if you can see, there are no photoreceptors across this gap right here. We don't have any of those layers in the gap. So there are no photoreceptors right there. That's why it's a blind spot. That's what the optic disc is. Fovea centralis, on the other hand, is kind of the exact opposite. It's a pit in the retina. And so imagine if light is coming in this direction, right? We want the most, the densest area of cones is in the fovea centralis. And so we have really acute vision right here. And so the ganglion cells get out of the way. Most of the bipolar cells get out of the way. So we don't have to go through those two layers of cells to get to the rods and cones photoreceptors at the back of the eye. That's what this pit structure is. That's what a fovea centralis is. And again, you don't have to know this histology. I just think it's cool looking. So here's a picture of kind of the anterior chamber of the eye, the cornea, the iris, the lens. The lens is held in place by these little spokes. Those are the suspensory ligaments. And the ciliary muscle. So the ciliary muscle has a ciliary body in it. The ciliary body equals ciliary muscles plus processes. So ciliary body is the whole big thing that includes the ciliary muscle and the ciliary processes. The ciliary muscle change the shape of the lens depending on if we're looking at things close or far away. And the processes create the liquid that circulates in this space, in that anterior chamber. The processes make aqueous humor. All right, and then I just have included some pictures of the muscles of the iris because you have some questions about them. So I liked this picture. So you have two directions of muscle. You have a direct one that goes around circular, and then you have one that radiates out like spokes. So understand the different jobs that those muscles are doing and understand the different parts of your autonomic nervous system that might be affecting those muscles. All right, so we're working through page 3 of our lab report, just answering questions about the structures that you just learned. Highlighting the tapetum lucidum here. This is a really interesting part of the choroid in animals. And so the video will kind of explain it, and then you'll be able to see it in some of my cow eye dissection pictures. So I've just included for you some cow eye dissection pictures from the interwebs. The video also goes over all these structures. So I just wanted to give you some study materials. So there's this picture. And then here's a beautiful labeled picture with all the structures. So the posterior aspect of the eye and the anterior aspect of the eye. And then here is a picture of all the pieces, parts kind of separated out. And so I draw your attention to this funky, like beautiful turquoise blue area of the choroid. This is tapetum lucidum. And it's responsible for the glow in your cat's eyes. If you see them out in the dark and they get flashed with something. Typically, our human eyes only have this black choroid. We want a photon to come into our eyeball, hit our retina once, and not bounce around, because we want a really clear signal from that photon or ray of light to transmit to a nervous signal. But animals who need to see in the dark, they need more than one chance at that photon. And so this area inside here that's all reflective allows for a photon to come in and then kind of bounce around a little bit and give more signals, more chances for that photon to hit a photoreceptor and be able to give information. So it raises the ability of animals, increases the ability to see in low light. So that's kind of cool. And then over here you have this kind of tissue paper thing. That's your retina, your choroid, your tapetum lucidum, your ciliary body, the iris that kind of surrounds the pupil is more flat, and the ciliary body has little spokes in it. So the ciliary body is the one with the spokes, and the iris is more flat. That's the iris. Iris, ciliary body. All right. And then here is our cornea. Here is the lens, the hard lens. It's harder when processed eyes, smooth, more soft in a human body, a live person. And then here's the vitreous humor over here, the big blob of jelly. All right, page 4 of your lab is where we talk about the visual pathway of the brain. And there are some questions down below. And so basically we're going to be analyzing this figure and then answering some questions about how the nerves are sending information. And so these are your optic nerves, right? They cross at the chiasma or optic chiasm. And so some fibers... Each eye has access to both left and right fibers. And so we're going to kind of understand how light comes in and affects that and how the images get processed. So there's two main distinctions here in the questions about vision and field of view. And so I'm going to discuss that a little bit. So vision has to do with what's happening on our retina. So if the medial part of our retina is being stimulated by light coming directly in, is that the same thing as field of view? Let's take a look. All right. So let's look at the right eye and let's look at this piece of the retina right there. And so if we imagine light coming in directly in a straight line, how would light enter the eye? From where would light enter the eye to hit this part of our retina? Let's just draw a straight line perpendicular out. So it seems that to activate the medial retina, we're actually, the light photons are coming from the lateral portion of our field of view. And so while vision is occurring, vision has to do with what our retina is experiencing. The field of view is opposite. Because remember, if you see an R out in the real world, it's going to look like this on your retina. And so that's where we get lateral light energy coming in and affecting our medial retina. So what's happening on your retinas is the vision, and where that light is coming from, where those rays are coming from, is your field of view. And then we're going to put a lesion somewhere in these optic nerves and see what happens. And so if we cut the optic nerve right here, we're going to have a different effect than if we cut the optic nerve right here or right here. And so that's what you're going to be comparing and contrasting. On Moodle, I will include kind of a written explanation for how vision and field of view are different from each other. That should help you. And I also do B as an example. So check this out. It's useful. It's on Moodle. Okay, and that's it for vision. So now we'll move on to hearing and equilibrium. Page 5 of your lab manual or lab report, I believe. And so it's going to kind of just lead you through the anatomy of the ear, external ear, middle ear, inner ear. And then on the bottom of the page, you'll start looking at and watching some videos about some hearing tests. And so we'll be looking at some hearing tests. So I'll come back to that. Your ear is divided into 3 main regions. So you have the external ear out here, which is all of the cartilage and all that. Remember what kind of cartilage? Elastic cartilage. And then so this is all kind of amplifying the sound waves and directing them into your external auditory canal. Another name for that is external acoustic meatus. You could use both names, either name, and I would mark it correct. So sound waves are coming in from out here, right? They're getting amplified and directed to go into our external auditory canal, where they're then going to bounce against the tympanic membrane or your eardrum, and they're going to make it vibrate. So then the middle ear, and this is all air-filled chamber. So this is an air-filled chamber. There's going to be something fluidy down the line, but we're not there yet. Then the middle ear is where the bones live. And so behind your tympanic membrane, we have this air-filled chamber where 3 auditory ossicles live. Malleus is the first and closest, the one that interacts directly with the tympanic membrane. Incus is the one in the middle. And then stapes is this tiny little stirrup that interacts with the inner ear. These bones are going to vibrate, and that stirrup is going to push in and create waves in some fluid that lives in this whole snail shell part. And it's going to push in at a structure called the oval window. Oval window. And it's a little membrane that allows the bone to push against it and cause waves in the fluid in the inner ear that are going to travel around, and then they're going to travel up to the top of this snail shell, and then they're going to come back down in a different tube of the snail shell, and they're going to bounce out against the round window. And this just allows the waves to propagate through that fluid-filled space. So, auditory ossicles, Malleus, Incus, stapes are in the middle ear, air-filled chamber. There's also a eustachian tube or an auditory tube or a pharyngo-tympanic tube that leads to your nasopharynx. And so this is the tube when you get on a plane and your ears need to pop, you hold your nose and you breathe out, and it's going to send some air up this tube and help equalize pressure on both the middle ear side and the external ear side of your tympanic membrane. And so ear infections are common in this little area and can drain into your throat. Then the inner ear is where we actually have our sensory cells. And so the inner ear is divided into 2 main areas. You have the pardon me. You have the cochlea, which is the snail shell part, and then you have the semicircular canals, which are these hoop-diddy-hoops. And so the semicircular canals have hair cells in them, or sensory cells, that deal with equilibrium and balance. And then your cochlea has hair cells in it that deals with hearing. So cochlea is for hearing, semicircular canals are for equilibrium and balance. All right, so back to your lab instructions. You have 3 videos to watch for the hearing and equilibrium tests. The first is the Rinne and the Weber test. So what's going to happen is the Rinne and the Weber tests measure conduction deafness versus sensorineural deafness. And so think about conduction deafness as sound waves cannot be conducted to my tympanic membrane for some reason. They can't get there. Sensorineural deafness has to do with my nerves are damaged or not working somehow. They can't send a signal. And so that's what these tests tell apart. The Weber test is going to come first, and then if that's abnormal, then you do the Rinne test to get more information. The Barani test has to do with spinning in a funky chair, and the Romberg test has to do with what happens to you when you stand straight and close your eyes and lift your arms. Do you lose your balance? And this link is the wrong link, but the correct link is in your lab report. So we'll see that in just a second. So here's your link for the Rinne and the Weber tests. And then some questions about the difference between conduction deafness and sensorineural deafness. I've left you some nice little color-coded helpful hints. Watch the videos. It should help you determine, what is normal hearing in a Rinne test. Sound conducted through the air or sound conducted via the bones of your skull. Then 11 and 12 have to do with proprioception and equilibrium, and so you can find information about those in your lab manual. And then here, the Barani test has that link, and the Romberg test. This is the correct link for the Romberg test. The lab instructions is incorrect. This one works. So watch the videos. All you really need to do is determine which test, which test goes with each, scenario. So which test has to do with equilibrium? Which test has to do with proprioception or balance receptors in your body? Which test has to do with determining if there's any conduction deafness? That kind of thing. You don't have to go into a lot of details about these tests, just kind of know which test goes with what. Okay, and that's it for hearing and equilibrium. So then we move on to taste, exercise 26 in your book. All you really have to do is kind of understand how the taste receptors work and where they live on your tongue. And so I've given you some information here about which papillae do not have taste buds, but I also found a great picture that I'm going to show you that helps to explain that. Then you kind of talk about, just read in your lab manual about kind of how the taste and smell receptors work, what is the importance of saliva, and then some kind of questions getting you to think about taste receptors. Do babies have really sensitive taste receptors, or do they have undeveloped taste receptors? You have to kind of think about that and ponder. And then kind of question 8 is all about the mixed senses working together to give you information. So I just gave you a slide with a bunch of pictures of taste buds. And so full of information. You don't have to know any of the histology pictures, like, these ones. You don't have to know them. I just wanted to include them so you could see what taste buds looked like and where they were. So you have 4 types of papillae here. So some sort of little bump on your tongue, right? One of them doesn't have taste buds on it. So looking at this picture right here on the left, try to figure out what type of taste buds those are, or what type of papillae those are. Hint the location of taste buds will help you. So just kind of interesting. Here's our little taste buds right here. They're so cute. Little globes of taste cells living on your papillae. All right, and that is it for special senses. Thank you very much.